now. All right. Good afternoon. Fred, I hope you are there because I've not heard your voice. I was hoping we could get the whole class, but since it's just the two of you, they would make do with a recording. If you have any questions as we go along, you can raise your hand and then I can pause and call you. Or if there are any issues, maybe you can't hear my voice clearly or there's something wrong with the screen, let me know so that I can quickly address those. While the class is in session, please, everyone should stay mute. And then if you have a question and I call on you, you can unmute and speak. So last week we met and we talked about tissue culture, plant tissue culture. I just want us to do a very quick recap of what we did. So we talked about what tissue culture is. We also mentioned the steps or stages in tissue culture. We talked about the lab setup, why it is important to keep things separate, the media room separate from where you actually do your culturing and subculturing, why it is important to do sterilization during tissue culture, and the types of sterilization that we have. We also talked about the plant media, its components, and some types of plant media as well. We mentioned the explant as a starting material for tissue culture, and there are different sources, as we mentioned. Each culture type uses a particular explant. We mentioned those as well. What else? Yes, we did mention that it's important your explant is fresh and young and less differentiated because it's much easier when you are doing tissue culture with those. I also provided some links that you can follow to better understand some of the topics that we have treated so far. We mentioned surface sterilization. We also mentioned moist heat and dry heat as part of the sterilization processes. We mentioned flame heat sterilization, filter sterilization, and then radiations. We also mentioned the basic requirements or the basic practices required for tissue culture. We also mentioned some plant hormones, which, did we do this part? Did we?
Hello, madam.
Yeah, William, did we do this part?
Madam, please, we didn't do that.
Okay, because I remember there was a part that I said you can read it yourself, so we can continue the next time we meet. I think it is the hormones bit. Okay, so I'll just walk through this slide and the next one. Yes, so that we can move on and do the topic for today. I did make mention of, yes.
I think one of the guys just sent a message to our group. I don't know, maybe if he's finding it difficult to join, let me check and see, so at least. That's Asari.
Okay. I haven't received
so maybe
anything, because if he's trying to join, I would see it here and then I can, but you guys were able to join without me accepting or admitting you. So I think it should be easy for everyone to join.
Oh, okay. Then let's continue.
Yeah, maybe it's his internet.
Oh, okay.
All right. Okay, so I did make mention of the fact that during tissue culture, differentiated cells are able to be regenerated into whole plants. However, they will have to be helped with the use of hormones, okay? Because as I mentioned, tissues or organs such as roots, shoots, and leaves will be able to regenerate into the same kind, which means if you take an explant of leaf, it should be able to be regenerated into leaf. But then with the help of phytohormones, you are able to manipulate them so you can have the formation of roots from a leaf, the formation of stem, and then a whole plant. Okay, but with the use of the meristem or with the use of the embryo or seed, you can easily grow it into a whole plant, even without the help of phytohormones. But then you need the required nutrients for such as well. So some of the major plant hormones that are used in tissue culture are ozons, cytokinins, gibberellins, and then abscisic acid, as well as ethylene, but ethylene is seldom used, okay? For the ozone, its purpose or the main role or function that it performs is to stimulate cell elongation, root initiation, and then vascular differentiation. I think the rest are there for you to read, okay? Let me move on to the summary, which has a summary of what each hormone is able to do. Before then, this is an example of how cytokinin and ozone are used in tissue culture. So generally, the ratio of the two hormones, being ozone and cytokinin, can determine the plant development. For example, when equal amounts of ozone and cytokinin are added to the plant media, you find callus developing. So when you want callus development, then you would go for equal amounts of ozone and cytokinin in your media. For the formation of shoots, however, you need to use low amounts of ozone to high amounts of cytokinin. That's when you can have the shoot formation. And then for the root development, you would rather go for the shoot formation. You have high cytokinin and then low ozone. That helps with the shoot development. And then for the root development is the vice versa. So high ozone and low cytokinin would help you develop roots from your callus, okay? So now the summary shows the function of each phytohormone. Here we have the functions of ethylene outlined here. It tells you what ethylene is. It is a gaseous hormone. It promotes fruit ripening, which I'm sure you know as agriculturists. Lateral plant growth. It also helps with root hair formation and then female flower formation. For ozone, the functions are also elaborated here. Same for the different kinds of plant hormones that we have. So you can access this on the slides that I've shared with you. Moving on to what we have to study for today. We are going to talk about plant regeneration. Please, if the link should go off, this is an unregistered Zoom, so we are limited to 40 to 45 minutes. So if it should go off, just rejoin using the same link. So what is plant regeneration and what's the difference between plant regeneration and tissue culture? Can anyone give it a go? Can you give me an idea of what the difference are between plant regeneration and tissue culture? What's the difference? Are there any difference at all? How are they similar? So any of these questions that you can answer is fine, however you want to approach it. Sorry. There are just two of you, William and Fred. So who wants to give it a go?
Okay, madam, this is William.
Okay. Go ahead.
And when we say the regeneration, it's like the same plant using either the parts is that either the part, either the leaves or the stem, to replant the same, or maybe to reproduce the same plant.
Okay.
In the regeneration, a bigger size or bigger material is used, but for tissue culture, only small part of the plant is required for replanting or for rejuvenation.
Okay. All right. Any more ideas? Fred, do you have anything to add?
Madam, I think for perpetuation of
You sound very faint. Can you speak up a bit? Hello, Fred.
Madam,
Yes.
please, I'm with you. Please, can you hear me?
Yes, I can hear you.
Yes, please. I'm saying that with tissue culture, it's the use of any part of the plant. It could be the stem, the roots.
William, I don't know if it's just me who can't hear Fred. Can you hear him? Okay, looks like it's my internet. Okay. All right, sorry about that. My internet went. Let's share the screen again. And then... So Fred, I missed everything you said because my internet went off.
Yes,
I don't know if you're able to repeat. Yeah.
I was saying that plant tissue culture is the use of any of the plant. It could be the leaf, the stem, the roots, to regenerate. I mean, to make copies of the same plant species, aseptic condition. Please, can you hear me?
Yes, yes, I could hear you. Thank you very much. Okay, so listening to the both of you, it's quite evident that there's very little difference between tissue culture and regeneration. It's kind of difficult to tell the difference between tissue culture and regeneration.
The difference.
Do you see any difference that you
Madam,
would have to share?
I will tissue culture. It's like in a form of regenerating plants.
Mm-hmm.
But then with the tissue culture, it is done with say it could be a part of the plant, and then I don't know whether I'm right.
I missed some parts, but I think I heard you. Okay, so what I would want you to know is okay, so what I want you to know is plant regeneration is a major, I'll say its major source is from tissue culture. Okay. Plant regeneration basis on tissue culture to be done. So it's the same techniques that we use in tissue culture that we use for plant regeneration. Is that okay? So as we go along, you'd get the picture of what I'm trying to say, but there's very little difference between plant regeneration and tissue culture. All right. So plant regeneration is a process of growing a whole plant from a single cell or a group of cells. Is that okay? It's also based on the principle of totipotency. And we've explained what totipotency is, which means the ability of a cell to be able to develop into its own kind. So,
Yes,
for
madam.
example, you put the
Yes,
embryo there and it'll be able to develop into a whole plant. It's as a result of its ability to property of totipotency, so to say. Please, can you mute yourselves because there's a lot of undifferentiated cells like the meristem, as I already mentioned, for differentiated cells, it's very, very difficult to use a stem, for example, to develop a whole plant. Without the help of phytohormones, it's close to impossible. But then for undifferentiated cells like the meristem or stem cells, and then embryonic cells, they are able to grow into a whole new organism. Okay. All right. So what is the difference between organogenesis and embryogenesis? You should know that plant regeneration can be achieved through these two different methods, which is organogenesis and embryogenesis. And their names kind of give an understanding as to what they are. So organogenesis is the use of organs for the plant regeneration, whereas embryogenesis would use the embryo for regeneration. Both of them have two different types, which is either direct organogenesis or indirect organogenesis. Same for the embryogenesis, it could either be direct or indirect. So what do we mean by direct and indirect? For the direct organogenesis, when you take an organ of the plant, it will be able to grow into a whole new plant without going through the stage called the callus formation. Okay, but once it goes through the callus formation, then it becomes indirect organogenesis. The same explanation stands for the embryogenesis. So when you have the seed and it goes directly to form shoots, roots, leaves without going through the stage of callus formation, then it is direct embryogenesis. But then if it has to go through the callus formation before the other organs are formed, then it is indirect embryogenesis. I hope we understand this point. If you don't understand, you can raise your hand and ask your question so I can clarify. Okay, I would move on then. So as I mentioned, regeneration can be achieved through either organogenesis or embryogenesis. There are three common pathways of the plant tissue culture regeneration. The propagation from pre-existing meristems, okay, which is either from the shoot meristem or the root meristem. And you can use the shoot culture or nodal culture for these methods. And we also have the organogenesis, which we've already explained as the formation of organs from differentiated tissue. Then you can also use what we call embryogenesis. And with the embryogenesis, it results in the formation of a whole plant using the embryo. And in this case, it can be either a non-zygotic embryo or a zygotic embryo. When we say zygotic embryo, what we mean to say is the male and female organs are able, the male and female cells, sorry, are able to come together to form a zygote before the process continues. For the non-zygotic, which is also known as somatic, you can have just the male cell or just the female cell being able to regenerate into a whole plant. Please, is there anything that you need clarity on at this point? All right, so I would go ahead. There are other technologies that you may find under regeneration. And one of them is called calogenesis. It's a type of organogenesis in which only adventitious shoot bud initiate into callous tissue. So when you have the adventitious shoot bud initiation taking place in the callus, then it is called calogenesis. Rhizogenesis is when the only adventitious root formation takes place. So it is more like the indirect organogenesis or embryogenesis, where you have adventitious shoot formation from the callus, being calogenesis. And then when you have just root formation from the callus, then it is rhizogenesis. Please, is it clear? Is that clear?
Yeah, madam. It's
Okay.
clear.
Okay. So what is organogenesis? Let's go a bit deeper. I've already mentioned it is the formation of organs. Okay. Organogenesis is the use of plant organs to generate a whole new plant in vitro. So with organogenesis, it's a type of tissue culture or a type of regeneration where you use the plant organ, which could either be the root, the stem, or the shoot or leaf, to be able to generate a whole new plant in vitro. And I've already mentioned we have the indirect and the direct. The indirect is the development of adventitious organs or primordia from undifferentiated cell mass, which we know is called callus in the tissue culture by a process of differentiation. So once the callus is undifferentiated, it has to go through differentiation to be able to form the different organs that we require to form the whole new plant. Okay, and that process is called the indirect organogenesis. For the direct organogenesis, you are able to form the different organs of the plant from the cells in the culture. You don't go through the stage of callus formation. So the callus formation is eliminated and you can form the different organs from the cells directly. Okay, so here we have A. This A picture that you see here is the disorganized callus, which is the undifferentiated callus cells. Okay, the B is shoot regeneration. So you are able to see the shoots forming from the callus, as you can see here. The C shows you root regeneration, and clearly this root regeneration is not from the callus because you can't see any callus within there. You have roots developing into roots, and that's root regeneration. And for the D, you have a single somatic embryo, which has also been isolated and can be used to regenerate into a whole new plant. Okay. So as I mentioned, organs will be able to develop into the same kind of organs. So if you culture roots, you are able to develop only roots. However, with the use of plant hormones or phytohormones, you'll be able to use these roots to generate a whole new plant. Same with the shoots, same with the leaves. Okay. The development of organized structures can follow one of these three pathways before you can get organized structures. That is from the callus. The callus is not organized. It's an unorganized structure. Before you can get it into an organized structure, there are some pathways that you need to follow. One is the shoot regeneration, which is based on a unipolar structure with a shoot apical meristem. Okay. You can also do the root regeneration, essentially a unipolar structure with a root apical meristem. And then we have the somatic embryogenesis, in which there is a bipolar structure. So I already mentioned with somatic embryogenesis, there is no zygote formation. You can either use just the male cell or the female cell to regenerate it into a whole new plant. The shoot regeneration, as I've already mentioned, can be done using the shoot apical meristem. The root regeneration can also be done using the root apical meristem. Okay. So with the use of the root apical meristem or the shoot apical meristem, you can be able to grow this into a whole new plant, which is organized because it has all the other parts of a plant. And we already know that using the meristem would also have an added advantage, which is the production of virus-free plants. Okay.
Madam, please, can you come again? That's a particular explanation.
Which one?
The last one, I didn't get that one clear.
The one that has to do with virus-free plants?
Yes, please.
Okay, so I mentioned last semester that if you want to generate virus-free plants, you can do so using tissue culture. And by that, what you need is the meristem, okay? The meristem can either be at the shoot apex or at the root apex. So when you want to generate a virus-free plant, you can use the shoot apical meristem for your culturing or the root apical meristem for your culturing. And you can develop a virus-free plant. Please, is it clear?
Yes, madam.
Okay, we'll go ahead. So now talking about the indirect organogenesis. Plant organ formation happens, but it has to go through the callus tissue formation. So you have your explant, you culture the explant, it would first form callus before you can find your the development of your organized structures like the roots, shoots, and leaves. Okay. There are some growth regulators that help with this kind of organogenesis. Yes.
Madam,
yes.
your network went off.
Oh, really?
Hello?
Yes. William. Hello.
Hello?
Yes, William, I can hear you.
And like we lost your voice. So if
Okay, so you want me to go over the indirect organogenesis?
yes, madam.
Okay, that's fine. I think we have about 1 minute and 20 seconds, and then it would cut. When it does that, just rejoin using the same link. Indirect organogenesis, as I've already mentioned, you have the plant organ formation, but then it first has to go through callus formation. So if you use your explants, you culture them on your media, you would first have callus forming before the structured organs are developed, okay? And I am saying that it depends or it utilizes some plant growth regulators for the differentiation to occur. So you have your organized structures, okay? We have the classic observations of Scooch and Miller that the direction of differentiation could be influenced by the ratio of exogenous growth regulators. So the example I gave earlier about ozone and cytokinin, when you manipulate the concentration of the ozone and cytokinin, you'll be able to direct how you want your undifferentiated cells to develop into structured cells. So if, for example, you require that you form shoots, you will have to adjust the concentration of